image do you interconnect with life? Is it this? This? How about these? Is life interesting? I mean, if you talk about it absurdly, life is just a bunch of chemical reactions and energy that is continuously occurring. Nothing's really interesting about it, right? No. Fortunately, life is very interesting and life is very worthy of our attention. Life, if we study it, or at least if we know something a bit about it, it can not only entertain us, it can not only pique our interests, but it can also be very beneficial for us. Greetings, everyone. My name is Gabriel Reano, and I've compressed my research into this 10 minutes or so speech, for I can share the wonders of life and how it was made possible to you. Today, I'll talk about reproduction and the molecule that made life possible, the DNA. Let's talk about reproduction first. Reproduction is our way of creating offspring that will become a part of the next generation, and that next generation will generate more offspring and more and more and more, so on and so forth. It's a cycle, and without the cycle, life would be impossible. In the floral kingdom, I've identified three ways in which plants can reproduce. The first one is pollination. It is the only sexual way of plant reproduction. This process starts when a male pollen gets transferred into a female flower or a female cone, and this pollen will then fertilize the egg inside the ovum. This will then create seeds. The second way is called budding. Budding is done when the parent grows a bud. The bud will then develop whilst it's attached to the parent, and when it is fully grown, it will detach and will live as an independent organism. The third way is called vegetative propagation. Vegetative propagation is basically done when a part of an individual plant, such as a stem or leaf, grows into another plant. There are four types of plants that use this method. The first one is bulb, second, tubers, third, rhizomes, and fourth, stamens. And then there's the extra one, which is called artificial methods, which is basically a copycat of vegetative propagation. Heading to the fauna kingdom, there are 6 ways in which animals can reproduce. 3 of them are sexual, and 3 of them is sexual. Let's start with the asexual one. The first is budding, which is the same as the earlier budding, but animals do it. The second way is called fragmentation, in which a sliced fragment of an organism grows into another individual. Starfishes are famous for using this method. The third one is called parthenogenesis. Ants and bees do this. It is done when a zygote forms without the need for fertilization. For the sexual ways, we have self-fertilization, which is done by hermaphrodites. Hermaphrodites are animals that have both male and female reproductive capabilities. Next, we have external fertilization, usually done in aquatic habitats. It is the exertion of male and female gametes in some place, but not always at the same time. It's just basically luck. And when the male and female gametes meet, they will fertilize. As the name suggests, it is done outside of the body. Lastly, we have internal fertilization, and it is the exact counterpart of external fertilization. In this method, the fertilization happens inside the body. There are 3 types of animals that use this method: viviparous, oviparous, and ovoviviparous. In a very simplified way, those are the ways of organism reproduction. For the next topic, we'll be talking about the DNA, the code of life. Most of us don't know why DNA is called the code of life. I mean, of course, it holds the genes or it holds the information about us, our traits, but it can't really be called a code if it's only like that. I mean, codes do have information, but codes also have commands or specific steps for some process. And yes, the DNA has it. Enter protein synthesis, the creation of proteins in the building blocks of life. There are two main parts of protein synthesis: transcription and translation. Transcription is the first part. It starts with the initiation. At this stage, an enzyme called RNA polymerase binds to the gene sequence called the promoter. The strands then unwind from the promoter region, which creates a transcription bubble. After this stage, the next stage starts, which is known as elongation. This process is basically the reading and the copying process of the information in the DNA by the RNAP. But because there's so little free time, I'll utilize this to explain nucleotides and the prime ends. Nucleotides is the base of DNA and RNA. There are 4 types of nucleotides, and they are represented by the first letters of their names. The 4 nucleotides are adenine, thymine, cytosine, and guanine. Nucleotides should always be with their specific partner. For reference, A and T should be together, similar to C and G. Primins, on the other hand, are the numbers 5 and 3, which represents the ends of a DNA strand. For this process, the mRNA reads from 3 to 5 and copies from 5 to 3. For the last stage, transcription, termination is just a signal to stop the process of reading. There are also other stages for eukaryotic organisms like capping, polydenylation, and mRNA splicing, but I won't be discussing it for now. The second part of protein synthesis is called translation. It is where the read information from the DNA gets used to create proteins. It starts with its own version of initiation. It begins by having the small ribosomal unit binding with a tRNA. Because of the amino acids, the tRNA is now charged. The bonds travel across the mRNA to activate a specific codon which signals the translation process. The initiation ends with the joint tRNA and a small ribosomal unit binding with a large ribosomal unit. The elongation stage of translation is the creation of the amino acid chain. This follows a very complicated procedure and will take a long time to explain. So even though I hate it, I will leave you with a bit of curiosity about this process. So let's just head to the next stage now, termination, the last stage of translation. This starts with the stop codon sending the signals to, stop the translation process. This results in the addition of water molecules to break the bonds between the tRNA and the polypeptide chain. The tRNA and the polypeptide chain are then folded, creating a new protein. That is how life is created, a very complicated, intricate process that made possible by a single molecule. Well, it's not a single molecule, it's actually many, but usually this is where I'll end the speech. But I have one more thing to discuss, and I just can't afford not to discuss it because it piqued my interest. So let's run it out quickly. Let's try to explain genetic engineering by editing the gene of a corn. So let's say that we are scientists hoping to create an insect-resistant corn. Before creating something, we need to think of something first, right? So, for the first step, we need to identify what traits do we want to add remove, or edit in a certain organism that we chose. For this case, we want our corn to have an insect-resistant trait. After deciding, we need to find a certain organism that has those traits. After searching for an organism, we will use the Bacillus thuringiensis, a soil bacterium, because of its insect toxic crystalline proteins. Next, we will copy the gene trait that we wanted. To copy a gene, we need to copy the DNA itself. The copying process starts by taking the DNA from a certain organism, which in this case is a soil bacterium, then putting it in a carrier, which is either bacteria or yeast. Every time that the carrier produces, the DNA is copied. Easy to understand, right? Now that we have the gene that the process requires, we need to find a way to insert it to the subject, the corn. Scientists do it by using CRISPR-Cas9, a customizable gene editing tool that helps us to cut and paste specific genes in DNA. CRISPR-Cas9 is also helped by certain RNA to identify particular chunks of DNA. After inserting the gene, we just need to grow the subject and observe it. In a very simplified way, in a nutshell, that's genetic engineering. And that's it. We've reached the end of our speech, and I hope that you've learned something. I hope you'll keep being interested in life and things about it, trying to learn and trying to gain knowledge about it, even though how absurd it is. To end the speech, I just want to remind you that whenever you feel down, always remind yourself that you are just a container for chemical reactions and energy, and so is everyone. So don't be hard on yourself. So that's it. Once again, I'm Gabriel Reano, ending this speech.